Thank
Many of
you.
you saw the little biographical sketch that didn't have much bio, had more science in it. So I'll give you just a little bit of background and then we'll get to the lecture. So Dr. Persidsky actually received his beginning of his medical training in, and I'll mess up this, but you can give me the correct pronunciation, Bogolomet, Bogomolet, National Medical University in Kiev, Ukraine, and also received his PhD in pathology at the Kiev Research Institute of Urology and Nephrology in Ukraine, Kiev, Ukraine as well. And then he stopped by for a 2-year fellowship in Strasbourg in France. All of this, of course, was in pathology, and came to the University of Nebraska Medical Center for his residency, where he stayed on and rose through the ranks to full professor and was recruited to Penn in 2008. Sorry, we'd love to recruit you to Penn, recruited to Temple in 2008, where he now is the chief of pathology service at Temple. He's the medical director of the clinical laboratories there, Temple University Hospital and Genes, and also pathologist in chief at Temple. Dr. Persidsky has a long history of research and highly published, highly funded career with north of 171 peer-reviewed publications. He served on multiple NIH peer review panels and basically has a career that would be an envy to all. And Temple recognized that and has basically appreciated him. We hope appreciated him enough. We basically are going to hear from him today about a career path that intersects with the interest of a lot of people at our centers. We basically are dually hosting Dr. Persidsky from the CFAR, the Center for AIDS Research at Penn, and the Center for Study on Addictions. And basically, there's a lot of interest in neuroinflammatory processes, and of course, there's a huge intersection of HIV and substance abuse pathology. And basically, even though Dr. Persidsky could give a number of lectures on many different diseases and disorders that have disruptions of the blood-brain barrier and that are therefore pro-inflammatory, we've asked him to talk to us today about the intersection of HIV, substance use disorders, and neuroinflammation processes. So Dr. Persidsky, please proceed. Thanks so much for coming to us today despite the snow.
Yeah, thanks a lot for invitation, and I think always humble when somebody introduced me so highly. So as you can see from my first slide is all of us, we are aware that despite of recovery of patients infected with HIV and well control of HIV replication, some cognitive impairment continues to be highly prevalent in this group of patients. And we strongly believe that it's because of low level of HIV replication in CNS, in reservoir cells like macrophages and microglia, and also because of the injury of blood-brain barrier mediated by pro-inflammatory factors in the blood and a low level of migration of cells into the brain. Evidence of blood-brain barrier injury exists very early on in infection, and it's proven to be by presence of pro-inflammatory factors and also leak of albumin across into CSF. So clearly, blood-brain barrier is being compromised very early on, and we always were, under impressions that treatment approaches targeting inflammation, blood-brain barrier, and most probably HIV replication altogether will be the most beneficial. So blood-brain barrier is composed of highly specialized endothelial cells connected by tight junctions that lay on a basement membrane, and immediately outside of them there are pericytes, the cells which we have personally started studying a few years back. And there are endfeet of astrocytes, and altogether this group of cells function together is a so-called neurovascular unit. So here is the schematic of these tight junction proteins, some of them transmembranous, some intracellular connected to cytoskeleton. And also another feature of blood-brain barrier is that endothelial cells express in highly organized fashion a number of transport systems, including glucose transporters, some amino acids, but also so-called drug-resistant associated transporters like PGP and BCRP, which impede significantly penetration antiretroviral drugs into the brain. So all days when there was very little hope in terms of suppressing HIV replication, neuropathology of HIV was associated with migration of monocytes activated carrying provirus into the brain and then establishing themselves and differentiating in macrophages where they start producing virus and interacting with other glial cells, produce neurotoxins which led to neuronal demise. We had some evidence early on that indeed this happens, meaning the disruption of tight junctions. And here is an example of some of from our old work when you can see that this mononuclear cells migrating across blood-brain barrier in the case of HIV encephalitis disrupts these tight junctions as the cells migrate in between of tight junctions. While in controls, you can see there is contiguous staining. We're able to demonstrate that these changes in accompanied if you co-culture monocytes, primary human monocytes with or without infection. With primary brain endothelial cells, there is significant activation of small GTPase Rho kinase. It coincides with the phosphorylation of tight junction proteins and disruption of blood-brain barrier. Here's a measurement of transendothelial cell resistance. And if you block this process by Rho kinase, you can prevent phosphorylation tight junction proteins, disruption of blood-brain barrier, and also migration of mononuclear cells across artificial blood-brain barrier. So things significantly changed since invention of or introduction antiretroviral therapy. And a few years back, studying parasites, we were interested in terms of what's happening now at the level of blood-brain barrier. And we used 23 cases of HIV-infected patients who died in our hospital with minimal, relatively minimal brain pathology, and look into in blue staining of parasites, and in red this is activated microglia. Maybe only one case remotely kind of reminded us something like encephalitis, but you can see that progressive decrease of staining of blue, and while there is increase in staining of this activated microglia, even in patients with art therapy. And so what we think is happening is that although there is little migration of cells across the blood-brain barrier, but there is definite decrease of tightness, chronic tightness, even in art-treated patients. And you can see that in here, this is a significant decrease of the staining of parasites. And as we think that parasites is part of this demise of blood-brain barrier. So in order to study disease in its current state, we adopted, most probably 6 or 7 years ago, a model of humanized mice. When this is a mice, NAD mice which are reconstituted at birth with human CD34 positive progenitor cells, and by age of 1.5 to 2 months, they essentially rebuild human immune system. And here is the data on engraftment. This is uninfected, this is infected animals, and early examples of dynamics over a period of 2 months, dynamics of CD45 human cells in uninfected animals and infected, and you can see there is a drop of CD45, essentially mimicking the decreases CD4 positive cells. Constant viremia in order of between 100 to 300 copies of viral copies in peripheral blood, and fluctuation of CD8 positive cells. But we will also be able to demonstrate that there is relatively little migration of the cells into the brain, although there are now some attempts to actually manipulate this as well, and human activated cells are seen in perivascular spaces or in intravascular, in the vessels, in meninges, in the brain parenchyma. But we were able to demonstrate that these animals which are infected, they had also similar decrease in blood-brain barrier disruption even without migration of the cells in infected animals. Here is in green staining for 1-uptigestion proteins Claudin 5 and for PDGF receptor beta, and you can see that in infected animals the staining is decreased. So essentially we are mimicking something which we observed in humans now in animals. Obviously, studies of polydrug abuse, including smoking, opioids, or other drugs, alcohol use disorder, very difficult, but they are really of importance because they're highly prevalent in patients living with HIV. High level of coincident tobacco use among intravenous drug abusers noted in literature. Majority of heroin abusers, also tobacco smokers, and clinical data indicates that over 85% of methadone-supported patients are tobacco users. And smoking overall is 3 to 4 times more prevalent in patients living with HIV, and is considered one of the major risk factors for cognitive decline in this population. So we use this NSG humanized model. Here is the overall design of these experiments. They were exposed to cigarette smoke over a period of 6 to 8 weeks. Here's control animals. Here's HIV infected, and they were and in parallel they were also injected by mini pumps delivering 1 milligram per kilogram per day of morphine for last 2 weeks of exposure. When we looked into peripheral blood, we showed that there was increase in, there was essentially all animals developed viremia, but there was no statistically significant differences in between different groups. Was variation in weight. We always pay significant attention to it because partially it's reflection of metabolism, partially reflection of intensity of inflammatory responses. And non-surprising, animals which were exposed in smoke with or without morphine lost weight. Unfortunately, our control morphine group alone was lost because of technical failure. Something happened so essentially all of them died from overdose. So fortunately, we don't have data from that group. What we observed in peripheral blood, there were changes in T cell profile in humanized mice. What we found that there was a significant decrease in level of CD4 cells, and they obviously were decreased as compared to controls, but combination with smoke smoke and morphine as compared to controls where CD4s were even more significantly decreased, and even in comparison to HIV, while level of CD8 cells increased. Therefore, ratio was even more skewed in CD4 versus CD8. When we looked into expression of PD-1, perforin, and granzyme B in T cells, we found that animals exposed to morphine, both PD-1 as well as granzyme B and perforin, they showed significant increase. And for us, although this is a markers of immune response, but all of us we are aware that most probably part of the problem in HIV infection it's overexpression, overreaction of immune system. And so we think that this signature of HIV infection was further enhanced by morphine, but not by other treatments. We looked into also expression of 29 cytokines and other biomarkers in peripheral blood. And to our surprise, only CCL13 was significantly elevated by HIV infection. There was also changes in TNF in smoke-exposed animals, and surprisingly, some cytokines were actually suppressed by HIV infection. So, in this regard, from this perspective, it would be difficult to derive any kind of coherent conclusion what happened in peripheral blood of these animals from this perspective. But again, this is a human cell, so it was relatively limited. When we looked into brain staining for microglia, what we found that there was that HIV versus control didn't increase significantly number of activity of microglia, but combination HIV with smoke and morphine and combination of both definitely increase microgliosis, and you can see even almost formation as a giant cells, and it's something which is reflective of neuroinflammation. Therefore, this combination we kind of proved to ourselves that we can do these long-term experiments looking into effects both opioids as well as, and this study we just published first part of the study, and we are right now working on peripheral blood, some markers with some exciting things related to monocytes, and the second one also eventually also some data related to lung. So few years back, one of my MD PhD students who was deciding between whether he should study effects of diabetes, which we are interested in, HIV versus electronic cigarettes, and finally he decided that he'll study electronic cigarettes. So it was a pretty difficult project because it's a combination of different types of e-cigarettes, their composition, and also comparison to tobacco smoke. It's a really tall order in terms of studies. On the left side, here's some in color-coded manner, some effects of e-cigarettes, electronic cigarettes with the effects which are nicotine dependent and independent. And if generally speaking, if you look that a lot of effects of e-cigarettes as well as cigarettes which overlap to extent, actually they're related to microvasculature. And the reason for us kind of start studying e-cigarettes were, first of all, is that they are highly addictive, but nothing is known about their effects on the brain, and nobody before actually studied effects of e-cigarettes on blood-brain barrier. So we expose these animals for 2 months to chronic 5 days a week for e-cigarette exposure, and we were able to demonstrate presence of significant amount of cotinine, it's byproduct of nicotine, which is comparable to one in the peripheral blood of people who are vaping. Out of number of cognitive tests, we showed that only one short-term memory was impaired in one group of animals, which are actually e-cigarettes without nicotine. We found that there is increased permeability of blood-brain barrier. We use small tracer like sodium, fluorescein, and 10 kilodalton fluorescent label tracer. We found that there was a decrease in tight junction protein occluding in e-cigarette exposed mice, and also in GLUT1 major glucose transporter. Here's the results in different parts of the brain, and here so for GLUT1. We found also upregulation and activation of microglia, several parameters of microglia, interestingly enough, in basal ganglia in these animals exposed to e-cigarettes. We isolated microvessels from these animals, and you can see this chronic exposure to e-cigarette resulted in major increases in pro-inflammatory factors, cytokines, chemokines, cell adhesion molecules, transcription factors, and others. We developed few years back, I think we published in 2015, this paper when we combine a so-called cranial window, when we can remove part of the skull of the animal and dura, and observe leukocytes labeled in rhodamine in microvasculature and superficial brain vessels. But we also added to it cannula, so we can actually create aseptic meningitis and encephalitis in very controlled manner. For example, if you inject TNF, you know that reaction will start to occur within most probably like hour and a half. And so after exposure for 2 months of these 2 e-cigarettes, we looked into adherent and rolling leukocytes in microvasculature. And here's this, and baseline this, and relatively few cells are present. And you can see there was, all these data were not significant. But when we injected TNF, we found that as compared to control, there was major increase in adherent and rolling leukocytes in e-cigarettes, in regular cigarettes, e-cigarettes containing nicotine, but the highest level was actually in e-cigarettes without nicotine. So all the studies, when we are talking about some effects on e-cigarettes, it's most probably we need to pay close attention to nicotine content, because this most probably plays a significant role in this regard. Currently, besides just looking in animal models, we do have three-dimensional models of blood-brain barrier in collaboration with Rowan University. They create in a matrix small vascular channels populated with primary human brain microvascular endothelial cells, and we can expose them to different noxious stimuli. In this particular situation, e-cigarettes with zero nicotine, with 1.8% of nicotine, and one of the diluents, propylene glycol. And you can see that two of these conditions showed disruption of tight junction proteins. The good thing about this model is not only that it's under flow model, so we can change flow and create pressure, which correspond to whatever level of microvasculature we would like to have, but also we can do measurement on transendothelial cell resistance and permeability. Also, we are using our traditional tools in terms of measuring transendothelial cell resistance just in static conditions, which we use for a number of years. And this is another example. For example, there was minimal disruption by one of the components, e-cigarettes, but in combination with inflammatory insult, which is interleukin-1, it was worsened. And the same applies to another component. So bottom line is that if you combine chronic exposure to some e-cigarette conditioned media, there is disruption of blood-brain barrier, and currently we are looking into further exploring what are mechanisms behind this synergistic effects. This is scheme for acute and chronic exposure, and we think that most probably driving force between electronic cigarette effects on endothelial cells, which we showed in vivo, is most probably reactive oxygen species in combination, some effects of nicotine. Interestingly enough, we haven't published this data, but acute exposure to e-cigarettes actually leads to tightening of blood-brain barrier in the similar, inflammatory models. For long period of time, my group was interested since most probably 2003, 2004 in effects of alcohol in the brain and specifically in the blood-brain barrier. And again, it's partially driven that alcohol effects in HIV-infected patients is definitely plays significant role. There is a high level of prevalence of alcohol abuse disorder in this group, and definitely there are conflicting data in terms of effects on the brain and some immune responses. Long time ago, we showed that actually alcohol metabolizing enzymes are expressed in endothelial cells here as the CYP2A1 and the ADH. There is upregulation of their activity, expression of protein, and you can block it by blocking alcohol metabolism in endothelial cells, or actually if you withhold alcohol for 2 hours from the media. It results in production of oxygen-reactive oxygen species. This particular situation shown is in nitrate tyrosine. When we look closely to primary brain endothelial cells stained for 3 tight junction proteins, ZO1, Claudin 5, and occludin, we notice that approximately for half an hour of exposure to 1.5 to 2 times of legal limit of alcohol in the media was formation of small gaps and changes in cytoskeleton, which we can block by blocking alcohol metabolism or inhibiting myosin light chain kinase. Here is the same data, kind of functional data, showing that drop in resistance after application of alcohol. And if you keep alcohol in the media, it continues to disrupt blood-brain barrier. And but if you withhold alcohol, it's actually blood-brain barrier seals itself back. And we have also corresponding data for permeability with FITC, dextran at 10 kilodalton, as well as that this compromise barrier actually enhances migration of primary monocytes across the blood-brain brain microvascular endothelial cell monolayers. And this data can be also reproduced in vivo. This is measurement of tracer 1.5 hours after infusion of alcohol in animal. It's about amount is 1.5 times of legal limit. We showed that similar to what we observe in interaction of HIV or infected or uninfected monocytes, the disruption of the barrier by alcohol accompanied by phosphorylation of tight junction proteins, and you can again block its occluding and clouding, and you can block it by blocking alcohol metabolism in endothelial cells or blocking myosin light chain kinase. And as indicator of that is myosin light chain kinase, which is responsible for this, is the amount of phosphorylated form of myosin light chain. We moved some of our also studies in vivo, and we used kind of prior iteration of this humanized mice. These mice were injected with PBLs intraperitoneally, and 1 week later they were injected in the brain with human macrophages derived from the same donors, infected with low level of this thing. This model is relatively short-lived, but it really represents kind of acute encephalitis with migration of CD8 cells across the blood-brain barrier. So animals were exposed for 4 weeks to alcohol, and they were sacrificed 1 week and 2 weeks after infection. What we were able to show that there was significant increase in viremia, partially driven by decrease of interferon gamma producing cells and diminished expression of immunoproteasomes in the spleen. When we looked into the brain, these are serial sections, showed that there is P24 positive HIV infected macrophages, CD68, migration of CD8s, and microglia activation. What was interesting, originally I thought that there would be fewer CD8 cells migrating into the brain of alcohol-fed animals, and this would explain. In reality, the numbers were the same, but number of HIV-infected cells in the brains of macrophages, essentially, in the brains of these animals were higher in animals exposed to alcohol, and we think because there was not efficient immune responses. This is why it kind of was more kind of profound presence of P24 positive cells. We also looked into microglia reaction, and we use just animals where macrophages were injected without any peripheral reconstitution and then exposed to alcohol, or they were reconstituted without or with alcohol, and there was progressive increase in microglia reaction. So essentially, there was not only immune responses drove this microglia reaction, but also in combination with alcohol, and partially it's most probably related to, again, here's nitrotyrosine, so we think there was partially related to this reactive oxygen species. At that time, we kind of, introduced this idea that there is, in alcohol abuse, there is neuroinflammatory responses. And it completely independently, several groups specifically working on effects of alcohol in the brain, specifically Fulton Cruz and his group, they demonstrated that indeed in brains of alcoholics there is significant microglia activation and overexpression of CCL2, which is a major chemokine all of us know in HIV infection in the brain. And here is some other evidence of pro-inflammatory effects of alcohol overall on immune and inflammatory system. So in order to find some other potential, tools to fight this neuroinflammation and effects on immune system as well as on blood-brain barrier, we turn our attention to agonists of cannabinoid type 2 receptor, and it was which are highly expressed on inflammatory cells, immune cells, but these agonists they don't activate or act on CB1 receptor, which are responsible psychoactive effects of marijuana. As I mentioned, CB2 receptor highly expressed on macrophages and in lymphocytes, macrophages and microglia, but mainly when they are activated. It has been shown before that CB2 agonists, they afford neuroprotection in animal models of stroke, multiple sclerosis, and Alzheimer's disease. And when we started, very little was known about their effects in HIV infection or in brain microvasculature. So first, we looked into expression of CB2 in brains in autopsy brains patients, control patients, and with HIV encephalitis. In control patients, what we saw were very weak expression of CB2 actually in the brain endothelium, and in full-blown encephalitis with microglial nodules, there was expression of some inflammatory cells, like microglia, macrophages, maybe some T cells, as well as on brain endothelium. Since this part of this project was driven by our interest in alcohol, we also examined brains of chronic alcoholics and demonstrated that there was overexpression of CB2 in brains of alcoholics as compared to controls, but there was no expression of CB2 in alcoholic brains in microglia. We moved to in vitro studies on primary human brain microvascular endothelial cells and showed that CB2 actually is expressed on primary human brain endothelial cells. This is CB2. This is one of tight junction proteins. This is overlay. And we also demonstrated presence of protein in protein isolated from membrane fractions of primary human brain endothelial cells or humanized cell line. What was also interesting is that not only we detected this expression, but we also were able to upregulate it by interleukin-1, by TNF, and by alcohol, meaning that it's actually inducible. We looked into effects of cannabinoid type 2 receptor agonists in a model of systemic inflammation. This is LPS injection, and we showed before that that in brain microvasculature, injection of LPS creates also adhesion of leukocytes to brain microvasculature without migration across blood-brain barrier. And we were able to show that there is treatment with cannabinoid type 2 receptor agonists substantially decreases adhesion 4 and 24 hours after, as well as decrease blood-brain barrier permeability. This results with two different agonists. Partially, this was driven by decrease of inflammation in brain microvasculature, this by immunohistochemistry, as well as by here's detection of ICAM1 in isolated microvessels from brains of these animals, and here's the data from densitometry. When we pretreated the endothelial cells, primary human brain endothelial cells, and let monocytes to adhere, we showed that pretreatment of endothelial cells with cannabinoid type 2 receptor agonists in dose-dependent fashion decreased adhesion of primary human cells. And it was driven, as shown here, on also via decrease of expression of adhesion molecules ICAM and VCAM on brain microvascular endothelial cells stimulated with TNF or LPS and treated with cannabinoid type 2 receptor agonists. We looked into changes in barrier tightness in vitro by measuring transendothelial cell resistance. Here's this barrier is treated with LPS, and here with ethanol, and you can see that this disruption was partially rescued by agonists. And another example on here is showing a few years back, which showed that there is increase of CD40 ligand in peripheral blood of patients, especially to extent you can correlate it with some cognitive impairment. And we were able to prevent this disruption by using agonists in this system. We think that partially it's related to their ability of CB2 agonists. It was shown by us first time is that there is that they have a potential of tightening blood-brain barrier without any inflammatory insult. And here is some kind of increase with two of them independently, and you can see we think it's associated with increased presence of tight junction proteins, including in claudin, in primary human brain microvascular endothelial cells. Real problem working with cannabinoid type 2 receptor agonists, they're very poorly water soluble. It's very difficult to administer. So we were very excited of finding 5 compounds, 3 derived from Roche, these RO compounds, and 2 from Leiden University, which are really water soluble. And here's some of the data just to show in dose-dependent manner increase of tightness of the barrier without any intervention after treatment of them with this agonist. They also anti-inflammatory. Here is the data when endothelial cells were pretreated with this agonist, and after TNF was applied, so you can see that if we treat endothelial cells, there is a major decrease almost to control levels of adhesion of primary human monocytes. So you can pretreat actually monocytes and you will have similar effect. And the same effects in most parts we observe also in migration when across blood-brain barrier models when we have chemotactic gradient created by CCL2. So there was a major decrease except one of them. Or you can pretreat monocytes and prevent their migration. We also demonstrated using the same inflammatory model when cranial window injection of TNF-alpha. And we showed that here's the baseline, the same vessels, and then after injection of TNF, there was major decrease in adherence of these leukocytes and this artificially created encephalitis, meningitis, and almost 50 to 60% decrease in adhesion. And here, this in terms of migration of cells was actually decreasing up to 90%, and we were able, in most parts, to decrease also blood-brain barrier permeability with all these compounds after systemic inflammation. We isolated microvessels from the brains of these animals and showed that between 32 to 46 genes were downregulated in endothelial cells stimulated with in microvessels isolated from animals treated with LPS, including MMPs, adhesion molecules, and tight junction proteins. So since this there is a potential of bringing this agonist not through intravenous injection. First time around, these drugs can be administered in the food. So we did this experiment using NSG mice, which were fed for 84 days, voluntarily fed with under control of technical personnel. We were fed with CB2 agonist 3 compounds from Roche. We used 2 different donors because otherwise we wouldn't get enough animals for one experiment. So this is why these results are shown for 2 separate donors. Despite that we showed before, and we have some data also to show that this particular agonist in vitro they decrease HIV replication, but it was not the case. In these experiments, all this data for 3 different agonists, and you can see that unfortunately we were unable to show direct effect on HIV replication. Here are the results on this weight, which we always record. what was interesting that we were able to show that all three agonists were able to rescue CD4 cells. It was really, they prevented decline of CD4 cells and also prevented this inverse ratio of CD4. Also, what was interesting, by using PD-1 and perforin and granzyme B in CD8 cells, all agonists in significant way decreased overactivation of CD8 cells, and and therefore overactivation of inflammation, which is not really, helps in terms of immune response, because all these animals have rampant viremia. And when we looked into the brains of these animals, this is standing for in blue for tight junction protein Claudin 5, in red for microglia. There was activation of microglia and decrease of Claudin 5 in these animals, and all three agonists were able to decrease activation of microglia to extend preserve also Claudin 5. So we think this is a very promising approach in terms of potential treatment, of HIV-infected patients. So just in summary, just talking about CB2 agonists, is that there are multiple places where they can be beneficial at the level of blood-brain barrier, at the level of monocytes, and we published this data and didn't have time to present it, as well as some effects of their neurotoxicity in the brain. So altogether, I hope that I was able to convey a message that we are dealing. So all these drugs of abuse, they're different, but surprisingly enough, we can learn something. For example, like with cannabinoids, that they could be actually protective and they can help in terms of this thing. We showed this data related to opioids, to alcohol, to methamphetamine, now tobacco smoke and e-cigarettes, and so definitely, something which is, these are very complex processes, but through combination of in vivo and in vitro approaches, I think we can actually really start to unravel basic mechanisms. And I would like to thank people of my lab, mentioned here, Tom Rogers, with whom we are collaborating on smoke and opioid things, as well as to Paul Patcher, with whom we are working for a number of years on cannabinoid type 2 receptor agonists, and people who generated these things, as well as NIH for financial support. Thank you very much for your attention. Thank you.
Thank you so much, Dr. Persidsky. That's quite a tour de force. And when we said working at the intersection of these disorders, you gave us not one but several disorders. It was really striking. I have a couple of quick questions, and I'm sure you'll have others. Early in the slides, you had a slide on microglia with both morphine and I guess nicotine, and it looked like it was almost a synergistic effect on microglia. Was it more than additive? Did it look synergistic to you?
By whatever we measured, it was kind of they were similar. I think
it's not critical. I just was curious about whether some of these things really, of course, they add up, but do they, synergize with each other and go off?
At least for a combination of two didn't increase significantly above what we showed with morphine separately and tobacco separately.
Right. That's interesting. And I was struck with the e-cigarette work because, of course, there's so much going on in the field with regard to, how to manage this very appealing modality for young people, and people still not being clear about what kinds of difficulties you have with e-cigarettes with nicotine administered that way. Could you say a little bit about polyethylene glycol as a culprit in any of this? Could you had that on one of the slides too?
Yeah. We in some situations, and these are relatively, some of this work is being done in vitro, because in e-cigarette, with these chronic experiments, we wanted to have real e-cigarettes, not just, so we'll need to repeat the same experiments in vivo, but it looked like there was a disruption or to extent of blood-brain barrier, at least in these 3D models. But definitely we'll need to work more on it.
Very exciting.
What is really people who are not working in the field, that it's you need to really to establish all the systems. You need to really to make sure that this is not extract of e-cigarette, just put in the media. This is really something which is coming out of jewel, this puffer through the media with making sure that, that we have amount of nicotine is exactly the same as we would expect in peripheral blood. So it's quite complicated system.
It is, but you had some elegant controls in there by looking at, zero nicotine versus more nicotine, and that was useful. The other thing I'll say, because part of the host today is from our Penn Yale NIDA P30 Center, which actually is using PET tracers and hoping to capture some neuroinflammatory processes, brain and maybe lung, but especially brain, in individuals with HIV and OUD, for example. Mm-hmm. And I was just curious, and there's probably many answers to this, but what is the effect on the entry into the brain of our PET tracers if you have a perturbed blood-brain barrier? Does it actually ironically increase uptake, or does it block uptake? Because sort of at a mechanistic level, we're always curious about what kind of uptake of tracer we'll have to be able to actually capture any effects. And so things have to make it past the blood-brain barrier, say that really fast. Could you comment a little bit about that? And I know that's sort of, you're not necessarily a PET researcher, but just about tracers getting into the brain well or not so well, or even more so.
My understanding is that the traces which are usually used as some kind of markers of activity or presence or activation of microglia versus some neuronal dysfunction, I think they readily penetrate. I don't have my own really true experience with that, because
that's okay. I just thought you might have a comment. Mm-hmm. And the very last thing is, I know that CB2 antagonists have also been looked at for neuropathic pain, and I know that you've done work with implications in that area. Would you want to say a brief comment about inflammatory processes and pain?
We are right now working on it, so it's truly too preliminary.
Really? Okay.
Too preliminary, and there are two aspects which we are interested in. Essentially neuropathic pain and potentially some neuropathic pain associated with also.
Very exciting with the therapeutics. That's really. I'll shut up and let other people ask questions, but as you can see, it was very stimulating for me. Other people, please jump in. Dr. Persidsky is here despite the snow. We have people on the call from the center who are clinical practitioners who deal with patients who often have more than one substance, right? They don't just have opioids or alcohol, but opioids, alcohol, and nicotine, and sometimes cocaine thrown in, and meth. We are nothing if not multifaceted when it comes to our patients' profiles of substance use. And you can imagine each one of those having potential insults on the blood-brain barrier. It's a kind of a scary prospect.
Yeah, one of the things that I haven't shown because of lack of time, and it's really preliminary, is now combination of e-cigarettes and alcohol, and is this inflammatory model on encephalitis. Combination of two actually led to exposure for 6 weeks to alcohol and e-cigarettes. Intermittently resulted in more pro-inflammatory changes and actually more inflammatory changes in microvessels isolated from this animal. So, but again, these are tricky experiments.
Absolutely, they're challenging experiments to do. Any other questions from folks? Don't be scared if you're not a pathologist or inflammatory. I'm neither.
My name is Mudit Tyagi from Thomas Jefferson University. So I was wondering how the CB2 activation affect HIV replication, transcription, etc. Does it enhance or it reduce?
It reduces. We published this in Journal of Leukocyte Biology in 2013. First author is Servio Ramirez. We showed that, interestingly enough, what we also discovered that maturation of monocytes versus macrophage increase expression of CB2 on them, and HIV infection increases even further. And we had this obviously confirmed in human brain tissue. So it decreases, and we showed that this current group of these agonists which we are using, we haven't published it there, but they also have suppressive effect. Right this second, I don't remember exactly whether we were able to show. I think it was something. I don't want just to speculate right this second. I just don't remember.
Thank you. Yeah, I mean, it's kind of makes sense, because I think inflammation goes down. I think NF-kappa B goes down. I think that's the reason that inflammation also goes down and HIV transcription also goes down and replication.
Yeah.
That is the link?
Yeah, and that particular work was done, like majority of what we are doing in vitro, in primary human monocytes and macrophages. So it was like we are using primary human brain endothelial cells. So we are trying to stay as far as possible from cell lines or something.